Hi folks, and welcome to another edition of Zog Science. Today we're going to be getting into cell respiration. We're going to lay the groundwork for that, talk about the process of glycolysis, and then talk about fermentation. So let's get into it. To begin with, living cells require energy. We talked about that before. Cells have to come back to the fact that entropy is always increasing, and because of that, they have to constantly be bringing in energy. The strategies for that differ for different organisms, but all organisms need to acquire energy. Remember that energy flows into ecosystems as sunlight. Most ecosystems, at least, there are a few that get energy different ways, but we're going to be talking about the majority of ecosystems, and then it is going to leave as heat. The process of photosynthesis, which we'll learn about next, generates oxygen and organic molecules, which are then used as fuel for cell respiration. Cells then use that chemical energy that is stored in organic molecules to regenerate ATP, which powers work. So ATP is actually used for organisms to be able to make work happen, to be able to do things. We're going to talk about the structure of ATP and how that happens during this video. Here's just a diagram showing that process. Light is coming in. It powers the chloroplast to do the process of photosynthesis that then produces organic molecules and oxygen. They will then go into the mitochondria, enable cell respiration to take place, which is going to produce ATP. ATP is going to power cellular work, and then we're going to produce some carbon dioxide and water that's going to be recycled in the process. So, first concept we're going to talk about is that catabolic pathways yield energy by oxidizing organic fuels. Now, I'm going to talk about redox reactions in this video a little bit, but you do not need to know redox reactions for our test, at least specifically. So, catabolic pathways, that's where we're breaking something down, involving the electron transfer. Our central process is the cellular respiration. So, electron transfer is a very important concept. Now, the breakdown of organic molecules is an exergonic process, meaning that we're releasing energy. And the fermentation is one of these processes. It's a partial degradation of sugars that occurs without oxygen. Aerobic respiration consumes organic molecules and oxygen and yields ATP, while anaerobic respiration is similar to aerobic respiration. It doesn't use oxygen. It uses some other thing. What we're going to be talking about here is cell respiration, including both aerobic and anaerobic, but we're going to be using it to refer to just aerobic processes. So when I say cell respiration in this class, I'm referring to aerobic respiration. Now, it is important to know that glucose, which is what you learn about in most biology classes as being used for cell respiration, is not the only molecule that can be used for it. But we're going to be using glucose as our model for how respiration takes place and understand that it's a similar but slightly different process for other molecules. So a redox reaction is simply the transfer of electrons from one molecule to another, and the transfer of these electrons is going to release energy that is stored in those organic molecules. And that released energy is then going to be used to synthesize ATP. So the redox reactions that we're learning about in this class are going to be taking electrons and moving them to electronegative molecules or atoms like oxygen, and that release of energy is going to be put to work. Again, you don't need to know specifically what a redox reaction is or what is being oxidized and what's being reduced in this class, but just wanting to introduce you to those terms and remind some of you that have taken AP chemistry about them. So during cell respiration, fuel is oxidized. Fuel would be something like glucose, and then the oxygen is reduced. So organic molecules that have an abundance of hydrogen, like carbohydrates and fats, are excellent fuels. And the reason why is that they have all these hydrogens that have electrons. And the hydrogens with their electron can then be transferred to oxygen, and the energy that is released can be used in ATP synthesis. So that's the reason why carbohydrates and fats are the best fuels for cells, for cell respiration. Now, in cell respiration, glucose and other organic molecules are broken down in a series of steps, and we're going to talk about those steps in this video and the next. Electrons from the organic compounds are usually first transferred to NAD+. So the electrons don't go directly from the molecule or from glucose to the oxygen. Instead, they go through an electron carrier. And the main electron carrier is NAD+. There's another one called FAD, which we'll get into in our next video. As an electron acceptor, NAD+ functions as an oxidizing agent during the cell respiration. So it accepts the electrons and it helps to transport them from one area to another. The NAD plus, when it is oxidized, when it is the oxidizing agent, turns into NADH. So it adds on a hydrogen and 2 electrons, and it is going to be storing energy that is going to then be used in order to synthesize ATP. So here's a picture of what NADH is, or NAD and NADH are. You'll notice that it is essentially a nucleic acid with some extra pieces, right? So here's the nitrogenous base. Here is the ribose sugar. Here's the phosphate groups. Notice it has 2 of them. And then it's got an extra ribose sugar and then another section over here. We can see when it is oxidized that we add on the hydrogen here with its 2 electrons in order to form that bond. Again, don't need to know specifically the structure of NAD plus and NADH, but you do need to know that it's a very important electron carrier that we're going to be discussing a lot. So NADH is going to pass its electrons to the electron transport chain. In the electron transport chain, electrons are passed down increasingly electronegative carrier molecules through a series of redox reactions. Those electrons are then transferred to oxygen at the end, and rather than doing it in one explosive step, we do it in a series of reactions. So if we take a look here, if you just take pure oxygen and pure hydrogen and you put them together, that is going to be an explosive reaction. They are highly volatile and they will form water, but they will explode when doing so if you provide a spark. But if we take those molecules and we control the release of energy, that is the bringing together of hydrogen and oxygen into water, then we can use that to form ATP, and that's what cells do. So what we're going to do is kind of preview the whole idea of cell respiration and then get into some of the more specifics. So glycolysis is the first part of the process, and it is going to be taking glucose and turning it into 2 pyruvate molecules. Then we're going to take the pyruvates and we're going to oxidize them and then put the product of that into the citric acid cycle, which is going to complete that breakdown. And then finally, oxidative phosphorylation is going to take place where we take all the energy that was released from the breakdown of the glucose and turn it into, and use it to form ATP. So here is kind of the outline of the process. We've got glycolysis occurring in the cytosol. Cytosol is another word for cytoplasm. Then you've got the citric acid cycle and the pyruvate oxidation. And then finally, you have oxidative phosphorylation. And we're going to go through each of those steps. In this video, we're going to talk about glycolysis. Now, oxidative phosphorylation is what accounts for almost 90% of the ATP generated by cell respiration. Most of that energy that is released is used there. However, there is some formation of ATP during glycolysis and the citric acid cycle, and it is done by what is called a substrate level phosphorylation. In this process, we have an enzyme that is going to transfer a phosphate group directly from a substrate molecule to ADP, which is why it's called substrate level phosphorylation. In oxidative phosphorylation, you just have a free-floating phosphate that is added to the ADP that's going to form ATP. And this would be an example of substrate level phosphorylation. You've got your substrate and an enzyme and ADP. The phosphate from the substrate is transferred over to the ADP, forming ATP. Now, this is the structure of ATP, and it's important that we understand the structure to understand how it carries energy and how that energy is released. Again, you'll notice that this is a nucleic acid. It's got an adenine nitrogenous base. It has a ribose sugar, and then it has 3 phosphate groups, which is why it's called a triphosphate. Now, the bonds between the phosphate groups of ATP can be broken down by hydrolysis. Remember, hydrolysis is the addition of water into a molecule. The energy that is released from ATP when the terminal phosphate bond is broken is the energy that can be used for the reaction. However, that release of energy doesn't come from the bond itself. It comes from lowering the free energy state of the molecule. So ATP has a very high free energy state. It's a very unstable molecule. It has a lot of energy pent up in it. When it turns into ADP, ADP is a much lower free energy state. And so because of that, that difference in energy is released and that energy can be used to do work. So the reason why ATP is such a high energy molecule is that it has these 3 phosphate groups. And you'll notice the 3 phosphate groups all have negative oxygens on them, and we know that like charges repel. So these are basically like all a bunch of negatives that are all bound together. They want to be apart from each other. So this is very unstable, very high energy. When you pop that phosphate off, there's a lot of energy in the molecule that is released, a lot of free energy that is released. Here's again kind of a picture showing that process happening. Hydrolysis occurs, we pop off this phosphate, we get the ADP, and then we get a lot of free energy that we can use to do work. All right, so let's talk about the process of glycolysis. Glycolysis is sugar splitting, so we're taking a glucose molecule and breaking it into 2 molecules of pyruvate. The net energy yield is going to be 2 ATP plus 2 NADH, and again, those NADH are going to be very important. That's where we're going to be making a lot of more ATP in oxidative phosphorylation. Glycolysis will occur whether or not oxygen is present. It is something that happens all the time. Now, I'm going to go through briefly the specific steps of glycolysis next, but this is what you really need to know. You need to know the big picture idea for glycolysis. You need to know that you start with glucose. You got to put in a little bit of ATP to start. Then you get out 4 ATP. So you put in 2, get out 4, which is why we have a net of 2. And we're also going to make 2 NADHs. And finally, we're going to be making pyruvate and water. Okay, so this is the big idea. This is what you need to know. And then I'm just going to briefly go through all the steps. So during the energy investment phase, what we do is we start with glucose. We add in our ATP. That adds a phosphate group. We then have another enzyme that changes the molecule. We then take that molecule, we add on another phosphate, and then we split that 6-carbon sugar into 2 3-carbon sugars. Now, I'm going to mention the G3P here because the G3P is a molecule that we will also see when we get to photosynthesis. Okay. Now we use an isomerase because these are isomers, these 2 molecules, and we're going to flip that all into the G3P. The G3P is then going to be used to make 2 NADHs. It's then going to be used to pull off the phosphates. So the phosphates that were attached are going to be pulled off, okay? And then we're going to produce some water, and then we're going to do the same thing we did before and make a few more ATPs, okay? So that's how the process of glycolysis takes place. Again, you do not need to know the specifics. You just need to know the general idea. We're ending up with our pyruvates, which are 3-carbon sugars, and there's 2 of them. All right, so now that we've talked about glycolysis, I know that the next step in cell respiration is the Krebs cycle and oxidative phosphorylation. We'll talk about that in the next video. I want to talk about fermentation because fermentation is glycolysis without those 2 steps, without the Krebs cycle and without oxidative phosphorylation. So we're going to talk about that process here. So anaerobic respiration is something that uses electron transport chain that is other than oxygen, okay? What we're going to be talking about is where we have fermentation. Fermentation is substrate level phosphorylation, just like glycolysis, but the pyruvate has to turn into something else, okay? So the first type is alcohol fermentation. This is where the pyruvate is converted to ethanol in 2 steps. The first step is going to release some carbon dioxide, and then the second step is going to result in ethanol. The alcohol fermentation is used by yeast, is specifically used in brewing, winemaking, and baking. The release of the CO2 is what carbonates beer. It also is what makes bread rise. And then the alcohol is what makes wine and beer alcoholic. In breads, it is going to be baked off, except for in sourdough breads. Sourdough breads are sour because there is still some alcohol left inside of them. Here is the process of the alcohol fermentation. You have glucose going through glycolysis, producing the 2 pyruvates. The NADHs are still produced, but those NADHs are then used to change the acetaldehyde, which is one of the intermediate steps, into the ethanol, and the ethanol is then able to be released. This also, again, produces the CO2, which is what makes the bread and yeast rise, or yeast bread rise. Second option is lactic acid fermentation. Lactic acid fermentation is done in muscle cells when there is no oxygen. So if there's no oxygen, you can't complete the citric acid cycle and the oxidative phosphorylation portion of cell respiration. And so you've got to get rid of that pyruvate somehow, and so lactic acid is produced. It is also done by some fungi and bacteria that is helping to make cheese and yogurt. So in this process, we go through, again, glycolysis. We produce the pyruvate. In this case, the NADH is used to change the pyruvate into lactate, which is then formed into lactic acid. Glycolysis is a really important pathway to know about, which is why we focused in on it today. It's the most common metabolic pathway among organisms on Earth. It's essentially done by all cells. Early prokaryotes probably generated ATP exclusively through glycolysis due to the low oxygen content in the atmosphere. And the location of glycolysis in the cytosol, again, the cytoplasm, indicates its ancient origins because prokaryotic cells have a cytoplasm, but they don't have a mitochondria. All right, that's all I've got for you today. Next time, we'll talk about the remainder of cell respiration. Thanks for watching, and I'll see you next time.